Good afternoon, everybody, and welcome to today's webinar. This is the first of several webinars this year on ensuring accurate classification of variants in BRCA1, BRCA2, and HRR genes. I've got a couple of housekeeping things to just go through first of all before we get started. So next slide, please. We'd preferably like you please to keep your microphones muted. We'll automatically mute them anyway, but please do not use your microphones until we get to the Q&A session at the end, which obviously like to avoid any background noise. We are recording this webinar as is normal practice, and we will make it available through our various media channels I think probably 2 weeks after this webinar itself. So keep a lookout for any comms related to that, and obviously you'll be able to dial into that and watch it back at your leisure afterwards. We do hope to get through to a Q&A session at the end. It's a pretty long webinar today. We've got a lot of fantastic slides from our speakers, but if you do have any questions, please do record them through the Q&A feature on Zoom. We will try and get through as many as we can at the end. Obviously, we've got 90 minutes, so we'll see how we go. But if we don't get through them, we'll obviously make sure that we include them in the report at the end of the year. So, sorry, at the end of the run. So don't worry too much if we don't get through to your Q&A, your questions this time around. We will get round to answering all the questions after the speakers have finished. Okay, next slide, please. Finally, so feedback is really important for us. We do listen to everything that everybody says. We want to make these webinars as good and as educational for you as possible. There's a lot of detail in the slides in the next hour and a half or so. So your feedback on how well we've done, what we could do better, etc., is really important to us. So we'll send you an email after the masterclass, which will have a link to a short feedback survey. Please, if you can, do spare the 2 minutes or so that it takes to complete that survey. And of course, we'll make sure we listen to that feedback and try to improve our activities moving forward. Okay, next slide, please. Okay, I'll hand over to you, Sandi.
Thank you, Simon. So I just add my own welcome to everyone to a new series of new webinars. Today we are joined by two of our expert advisors on the panel, and that's Miranda Durkie from Sheffield in the UK and Trudi McDevitt from Dublin in Ireland. And I'm Sandi Deans, Director of GenQA, and I'm chairing today's session along with my colleague Simon Patton, who's CEO of EMQN. So next slide, please. Disclaimer here. Thank you very much. That's the official part that we disclaim. And I'm now going to hand over to our presenters, Miranda and Trudi. We've got a great presentation for you today with a lot of helpful educational content. So we're going to move swiftly into the science side. And we have listened to feedback. We've been asked to go into more depth of some of the tricky variants. And we've extended this webinar from an hour to an hour and a half now. So we do respond to your views. So Trudi, over to you, please, for your lovely presentation. Thank you.
Thank you very much, Sandi and Simon. So we were just saying at the start of the webinar that we've now completed 9 GTAC runs and we're about to go into run 10. So we thought we would take this opportunity to look back through the previous runs to identify potential areas that might have been challenging to participants. So in this webinar, Miranda and myself will focus on some of these challenges, including classification codes and variants, particularly where there was a different outcome and where the outcome could have had a clinical impact. We're also going to just briefly review the BRCA1 and 2 and HRR gene guideline, variant interpretation guidelines that are currently available for participants to refer to for run 10. So next slide, please. So in our look back then, what we did was we looked for outcomes, for cases where outcomes were different to the expected outcome, and where 25% or more participants returned a different outcome, where there was a clinical impact in terms of treatment decisions. So for example, where the expected outcome might have been a 5 or a 4, but we saw a 3, 2, 1 and vice versa, and also where there was no clinical impact, covering classification codes 321 and 45. We also looked to see where participant returns were the same as the expected outcome, but where the incorrect criteria or strength had been applied. and also reviewed participants' questions for recurrent themes that might indicate potential areas where there might be challenges. So next slide, please. So for what we looked at was, as an example, in run 9, you can see 6 variants that were assessed during run 9. So the variant numbers are along the x-axis and the classification codes on the y-axis. So you can see the first example, the expected is in green, and the blue, observed were in blue. So you can see there's a difference there, and a 3 and a 4. So that would be considered an outcome with potential clinical impact. The other variants in the run, the outcomes were different to expected, but they were in between the classifications that wouldn't be really considered to have a clinical impact. So you'll notice the red arrow for clinical impact, and then the black arrows across the top. They should, all 5 variants should be black arrows. That's just an error there with a cross. They're all the same. So next slide, please. So what we found then, this is just a summary of our findings. We looked at runs 4 to 9 over years 2020 to 2022. You can see the number of participants who completed and the genes that were analysed in each run. So what we found was the numbers of outcomes that were different to expected are the first numbers in the final column. And of those in brackets are the outcomes where the differences could be associated with clinical impact. So next slide, please. The recurrent challenges that jumped out to us were in these main categories that are listed in the first column. So PVS1, which variants were appropriate for this code to be applied and which strength. Functional studies, which functional studies were appropriate to use and at which strength. Population control data, in particular BS1 and PM2. So for the cases that you're seeing on NOMAD, which of these criteria are appropriate to apply? The PS4, taking a case counting or case control analysis approach, which studies were appropriate and how to do them? Applying multifactorial data, which codes apply and which strength? In silico evidence, which tools and which strength? Assessing conservation for the codes BP1, BP3, PM4 and BP7, how to assess if a variant or area was strongly or weakly conserved. And then combining codes, which codes should not be used together, and the final adding up of codes to get the final classification. So Miranda and myself will look at each of these areas in a bit more detail. So next slide, please. So reasons for the different outcomes. Well, it's due to different guidelines being used, different tools and databases being referred to, inappropriate classification codes, appropriate classification codes but inappropriate strength, and then combining errors and final summation errors. So next slide, please. So which guidelines? At this point, we'd like to refer you to a really good webinar that was at the start of Run 8 that is a fully comprehensive overview of guidelines, databases and classification codes by Kathleen and Andrew and Miranda. It's available on the GenQA and EMQN YouTube channels and is a very good starting point for going into Run 10. So next slide, please. The guidelines that participants are currently using can be seen by these graphs here. So between run 6 and 9, you can see that the majority of participants are still only using the original ACMG 2015 guidelines. That's indicated by the blue section here. The dark orange section are those participants that are using both the 2015 ACMG guidelines and the ACGS 2020 guidelines that are based on guidelines for laboratories for interpretation in rare diseases. And in the grey section here are participants using those 2 guidelines plus the general cancer guidelines from 2020 by the CanVig expert group. There's also a section in yellow where participants indicate they're using other guidelines, but we don't know as yet what these guidelines are. So next slide, please. So in runs 8 and 9, that was last year, we still have 41% of participants using the ACMG guidelines as their only reference for variant interpretation. Just to highlight that there is going to be an update to these guidelines in 2023. Next slide, please. So guidelines that have evolved from the ACMG original rules. are the ACGS guidelines in 2020, designed to provide laboratories with practical guidance on how to apply the ACMG rules to rare diseases. And then very closely aligned to that and published initially in 2020 are the CANVIG General Cancer Susceptibility Genes guidelines that are very closely aligned to the ACGS guidelines, but with adaptations to account for the fact that the cancer susceptibility genes are associated with common phenotypes. Then we have gene-specific guidelines evolving from these for CHEK2 and BRCA1 and 2 from the CANVIG group, from ClinGen VSEP group for ATM, and then this year there will be the Enigma BRCA1 and 2 updates and PALB2 also due in 2023. So lots of changes expected this year. Next slide, please. So guidelines evolve as new recommendations come out, as occurred for the ClinGen SBI recommendations for PM3, PM2, PP5 and BP6, as the scientific literature changes with new studies, improved in silico tools, and also very importantly from feedback from diagnostic laboratories as more and more testing and more genes are being analysed. You can see an example here of an update from 2.16 to 2.17 from the CANVIG General Cancer Guidelines. And then to the right of that, you can see the reasons why the guidelines were updated. So there's a constant feedback process going on with new information being fed into the guidelines, because it's really important that the guidelines reflect current practice in diagnostic laboratories. So next slide, please. An example of active feedback into the guidelines can be seen here. So there were some questions from participants in Run 8 where it was clear that we needed some additional clarification needed to be added to the wording in the cancer susceptibility guidelines document. So there were some questions on seeking extra clarification on how to assess conservation and also how to assess REVEL scores for application to PM5. So based on this, the wording was made a little bit more clearer in the updates for the latest guidelines that came out in December 2022. So please do keep the questions coming, because it just helps us make the guidelines as user-friendly as possible. So next slide, please. So we're going into Run 10, which guidelines are available for participants to refer to. So the flyer that was sent out has advised that participants should apply the ACMG variant classification guidelines in accordance with any gene-specific recommendations for the cancer susceptibility genes where these exist. So either from CANVIG or the ClinGen SBI groups. There are new guidelines coming out in 2023, but as we go into Run 10, the guidelines that are currently available to refer to are for BRCA1 and 2, the gene-specific guidelines from CANVIG, for ATM, the ClinGen VSEP guidelines for CHEK2, for the CANVIG group guidelines. Both of these are available on the CANVIG website. And for RAD51C and D and PALB2 pending gene-specific guidelines, the general cancer susceptibility genes guidelines are the ones that are available to refer to. So now I'm going to hand you over to Miranda for the start of the exploration of the challenging classification codes. Next slide, please.
Thank you so much, Trudi. That was a great kind of overview of everything. And now we're going to start on quite a deep dive into those kind of tricky classification codes. And as Trudi said, particularly focusing on areas and variants that the classification differences could potentially lead to a clinical impact. So next slide, please. So I'm going to start with PVS1. So PVS1 is the code that we use for a null variance. This could be a nonsense, a frame shift, a canonical splice site, or an initiation codon, or a copy number variant, obviously where loss of function is a known mechanism of disease. And a lot of people think that PVS1 is quite an easy code. Actually, there are loads of complexities. So next slide, please. So my first example is from run 6. This was a BRCA1 variant where you got a single nucleotide deletion causing a frame shift and then a stop codon 68 amino acids downstream. And come back to that. So you can see from the histogram that the assessors were expecting participants to get a classification of VUS or uncertain clinical significance. But in actual fact, the vast majority of participants scored this variant as lightly pathogenic or pathogenic. And as we all know, that could have significant clinical impact for those if this was a real patient. Next slide, please. The second variant, it's 2 runs later, RAD51D, very similar. It's a single nucleotide duplication, again causing a frame shift. This time, the majority of participants did get the expected classification as VUS, but we've still got 75 participants scoring it as lightly pathogenic. Next slide, please. So let's go back and look at the PVS1 guidelines. So obviously there was some limited information in the Richard et al. Publication in 2015. And then in 2018, Tayoun et al. Published these PVS1 specific guidelines with a lot more detail and really helpfully this fantastic PVS1 decision tree. Next slide, please. So we're just going to look at part of that decision tree and in particular the part that's relevant for these 2 variants that we're looking at. So it's in both cases they were a frame shift variant, and so your first decision to make is whether this variant is predicted to undergo nonsense mediated decay. So on the right hand side of the slide is a very simple kind of schematic showing whether how to predict whether NMD might occur or not. So on the left hand side of that schematic, it's showing you where you get a stop in the last exon of the gene, and actually you have the ribosome is able to displace all the exon junction complexes, and therefore that RNA would be translated into protein. It might be a truncated protein, but it would still be present. However, on the right-hand side, it shows you if you've got a termination codon further upstream in one of the preceding exons, then the ribosome can't displace those EJCs and you triggers the nonsense-mediated decay pathway, and you get complete degradation of that RNA, and then you get no protein produced. Next slide, please. So there's a fantastic tool available on Decipher website. It's an absolutely brilliant, really easy to use website. So if you go to Decipher and then go to the protein tab and kind of select your gene of interest. And as we saw from the previous schematic, it's not quite as simple as it's exons in the variants in the last exon that escape NMD. Actually, nonsense variants or truncating variants in the first 100 base pairs, or the last exon plus the 50 base pairs of the penultimate exon can escape NMD. So I've just zoomed in on each of those areas, and it's a bit small, but you can see that there is this predicted NMD escape tract on Decipher, and you can see from that for BRCA1, NMD escape is predicted from codon 1806. Therefore, our variant from run 6, which has a frame shift starting at 1854 and then a very long tail kind of until you get a stop, there would be no nonsense mediated decay predicted. Next slide, please. And very similar for the RAD51D gene. Again, zooming in on that last exon, you can see that NMD escape is predicted from codon 284. And so our variant from run 8 with a frame shift from 322, again, would be no nonsense mediated decay predicted. Next slide, please. So the next step in the decision tree is to assess what the functional role of that region of that protein is and how much of the protein is involved. And in both these cases, if you zoom in on Decipher, you can see that both the arrows show where the frame shift starts, and not where the truncation is, but where the actual frame shift starts. And you can see in both cases, it's right near the end of the gene. You can see the kind of the X for the stop codon. And you can see that in both those cases, they are not functional domains, and they are very poorly conserved. And particularly for BRCA1, you'll notice that 68 amino acids downstream actually means that this variant is a protein extension variant, which I'll come back to. Next slide, please. So let's go back to our decision tree. So we know it's not predicted to undergo nonsense mediated decay. We know that the role of the protein function is unknown. It's not in a functional domain. We know that loss of function variants are not common in population studies. We can see that the variant removes less than 10% of the protein. Therefore, the maximum strength we can apply PVS1 for these variants is PVS1 moderate. Next slide, please. So now let's have a look about how participants applied PVS1. So remember, in both cases, class 1 is our expected classification. So BRCA1 at the top for class 3. You can see a smaller number of participants applied PVS1, and when they did, they tended to use it at the strength moderate. But you can see moving into class 4, a much higher proportion of participants applied PVS1, and they tended to do it at either strong or very strong. And then moving into the class 5, the majority applied it at very strong. And this was where these kind of problems in classification is coming from, by overweighting that PVS1. And we can see very similar for RAD51D, that the class 3 expected, a smaller proportion applied PVS1, and they tended to apply it at moderate strength. Those that classified it as class 4 more used PVS1, and they tended to apply it at strong or very strong, so overweighting it. Next slide, please. So a quick kind of add in for PVS1 versus PM4 for stop loss variants, because I think particularly we know we've got participants from all over the world, which is fantastic. So if you are looking just at the Richards et al. Guidelines, that there are some further developments in the ACGS 2020 guidelines that are available online, and particularly this decision tree for stop loss variants to decide whether you have non-stop decay, NSD, is very helpful. So I thought I'd point out that is available. Next slide, please. And just to quickly show you why this may be important, on the left-hand side is the schematic kind of showing the mechanism of non-stop decay. So if the ribosome runs all the way through and through that poly-A tail, you end up with this very long RNA that's targeted for degradation. And so if you have a stop loss variant, it's really important that you have a look at that 3' UTR to assess whether you have got an in-frame stop codon within that 3' UTR or not, which means you need to know what are the key signals you need to look for. So particularly the poly-A signal, the poly-A site, and then the downstream element. So this is a lovely example from James Drummond of the MLH1 gene. I know not relevant for HRR, but that you can see the poly-A signal, the poly-A site, the downstream element, and then you've got an in-frame TGA. So if you had a stop loss variant in this gene, you would not got NSD. And the reason I'm showing you this is because this is also very important for that BRCA1 variant from RUN6, because that was a protein extension. And again, it was really important that you checked whether there was NSD present or not, in which case there wasn't. If there is NSD present, you would get a null variant, and you would be able to use a strong code for that. Next slide, please. Okay, so I'm sticking with PVS1, but now I'm moving from kind of truncating into splicing. And this was a very interesting variant from the last run 9 in the ATM gene, and it's a +2 C to T. So we were expecting a classification of class 3, but actually we had over 150 participants scored it as class 4 or class 5 with those clinical implications. Next slide, please. So the top picture is schematic from Alamut Visual Plus, showing the splicing predictions. And hopefully you can all see that actually the only tool that predicted that natural splice site was Splice Site Like Finder. None of the other tools predicted this kind of rarer natural splice site. And the reason is, you can see the picture on the bottom, is that the most commonly used nucleotide at that +2 position is T. But actually a +2C is used in 1.4% of natural splice sites. So in this case, next slide, please. You can also looking at some of the other in silico tools. Splice AI showed no impact. And then from the other Alamut Visual tools, again, Splice Site Finder, found the natural splice site and so predicted no effect. The others, because they didn't see the natural splice site, that's an effect predicted. Next slide, please. So in conclusion, this variant changed a rare natural GCAG splice site to the very common natural GTAG splice site. We would predict that all the same spliceosome machinery and everything else is in use. It's still the major spliceosome. Therefore, just splicing is likely to be a bit more efficient and it's not going to affect gene transcription. So we wouldn't use PVS1. And this is actually supported by the ATM VSEP specific guidance, where they actually specify this variant and say to use PVS1 not applicable for it. Next slide, please. So looking then at the participants' application of PVS1, on the left-hand side, the class 3 expected, you can see the participants didn't use PVS1, but when you're moving into the class 4 and class 5, the majority of participants did use it and tended to use it at that very strong strength, which really would not be appropriate for this variant. Next slide, please. Okay, now I'm going to flip it on its head and talk about splicing, but +2 T to C. So in this case, where it's changing the common natural GT splice site to the rare natural GC splice site, which may still be functional, as we've seen from that previous example. And a really good example of this is the BRCA2 C.8331 +2 T to C variant, which was previously reported as pathogenic in multiple cases, but actually this study by Nix et al. 2022 was published, and they did a lot more work on this, and particularly RNA studies, and showed that this variant actually produces 62% wild type transcript. So that's shown kind of on the infographic on the left, and then in the middle they also did some family history weighting algorithm, and you can see from the blue line that the family history scoring actually fits right in the middle of benign controls. So this variant was then down classified to VUS or lightly benign. And I really, really highly recommend that you use either Splice Site like Finder or Splice AI is a fantastic tool for looking at these +2 T to C variants. And actually based on that, the ATM guidelines have looked through all their +2 T to Cs, and where there is some evidence from Splice AI or in kind of one case, some RNA studies, they've recommended that you don't use PVS1. I should say this is applicable to all genes, not ATM only. Next slide, please. What are some of the other complications to do with splicing? Well, you can get splicing, but the splice effect may be in frame. So I've got two examples here. On the left-hand side is the BRCA1 C.301 +1 G to A variant. And we've recently issued a down classification alert across the UK for this variant. You can see from the picture on the left-hand side that there is the natural splice site, but there is also a cryptic in-frame splice site that would delete 3 amino acids. And actually we've had some updated functional studies that have shown now that this transcript is functional and is not causing a loss of protein function. Another way that it can be is you could get skipping of the whole exon, but that the exon could be in frame, as we can see here for this BRCA2 example. So affecting a variant affecting the donor splice site of exon 2. You would get loss of exon 2, but this is in frame, and actually that there is no functional domain associated with exon 12. It's functionally redundant, and there are alternative transcripts without this exon. So again, PVS1 would not be applicable in either of these cases. So be careful when you have the +1, +2s. Don't just apply PVS1 without really having a kind of a closer look. Next slide, please. The other thing that sometimes participants go wrong is using PVS1 for the non-canonical splice sites. So here is an example here for -5, BRCA2 variant, and majority have scored it as expected as class 4, but we still have got quite a spread from class 3 to class 5. Next slide, please. And if again we look at PVS1 by participants' use, so for the class 4, you can see correctly that participants have not used PVS1, but when you go to the class 5, many participants have applied PVS1, which you should only be used for canonical splice sites. And then looking at the class 3, the major difference here was that there was no PS3 used, and actually there were a number of RNA studies available in the literature that showed that there was an effect of splicing. And that leads me very nicely on to my next slide, and the next codes we're going to talk about, which is PS3 and BS3 for the functional data. And the really key thing for these codes is that your functional study must be well established. Next slide, please. So my first example for this is a PALB2 missense variant, P.tryptophan 912 to glycine. So the assessors expected the classification to be class 3, which the majority of participants did, but we've still got over 120 classifying as class 4 and class 5, again with those clinical implications. Next slide, please. So what guidelines are available for PS3 and BS3? So we've had these very detailed, very good guidelines published by Brunich et al. On behalf of the ClinGen SVI in 2020. Next slide, please. And again, they've done this fantastic decision tree to really take you through, you know, all the different steps that you need to assess whether the assay that you're looking at is a robust, well-validated, good assay that you can score. So once you've kind of gone through the steps like 1 to 3, then you can do some statistical calculations to calculate your odds of pathogenicity based on the total number of benign and pathogenic variants that were included in the study. Next slide, please. And in the supplementary data, they have this odds path calculator available as an XLS that you can just download. So it's kind of pre-filled with lots of examples of how many pathogenic and benign variants are required to get different scores. And it's all color coded, so you can see the other odds of pathogenicity and converting that into the strength of PS3 and BS3. But what's really good about this calculator is that you can put your own data in. So for example, near the bottom of the slide, I've put in the data for the Findlay et al. 2018 saturation genome editing assay for BRCA1, and you can see that that comes out as red, and the green, which is a PS3 of strong for both, and the BS3 is strong. I've also put in the Boonen et al. Assay 2019, which is an assay for PALB2 that was commonly used for this other variant. And you can see that that comes out as orange, which is PS3 moderate. Next slide, please. So when we look at expected versus observed, you can see for when the class 3 as expected, a smaller number of participants have used PS3, and those that have tended to use it at the reduced strength of moderate. Whereas when we move to those that classified it as class 4, a higher proportion used PS3, and the majority of them applied it as strong or even very strong in some cases. Now, this was a very interesting variant that the assessors had quite a bit of discussion about, because actually the Boonen assay was validated using 20 pathogenic truncating variants plus benign controls, and they didn't have any pathogenic missense variants in there because they are extremely rare. And so we had discussions with the ClinGen VSEP authors, and they said that this time they're not including PS3 and BS3 in their classification. So we would actually recommend not using this for PALB2 currently. Next slide, please. So some good news is that lots of this work has already been done for you. So if you go to the CAMVAR website and the link is in there, and you click on the button saying functional studies, thank you, then it brings up this XLS, and these are all studies where the kind of CAMVIG steering advisory committee have already gone through some of these assays, included lots of detail, and then given you the strength of PS3 and BS3 that you can use. Obviously, if it's not on there, then you would have to do the calculator for yourself. Next slide, please. And again, we're very lucky in particularly in the BRCA genes, particularly BRCA1. We've got the 2 very large studies by Findlay and Bowman, but there are some variants that overlap in these. And so we wanted to have some guidance on how you would combine them, particularly if one said intermediate, one said loss of function, how would you score PS3 in those circumstances? So there's guidance on that in the CAMVIG guidelines as well. Next slide, please. So moving on now to PS3, BS3 for splicing. So that Brunich publication is only for protein assays. There is a ClinGen SVI group working on some guidelines for splicing, but it's not available yet. So meanwhile, we need to apply the same principles for any RNA splicing assays. We need to make sure it's a robust, well-validated assay. So has it been independently replicated? Were sufficient normal controls used? Is the splice isoform you've detected naturally occurring? Is the splice effect leaky? So do you get some wild-type transcript produced from that same allele? A reminder that you don't use this code if it's a canonical splice site. You would use it. You use PVS1 in those cases. And because a lot of the RNA studies often confirm the predictions that the resulting kind of transcript is out of frame and therefore would be degraded and be a null allele, some VSEPs are now using PVS1_O for observed for splicing assays. In the CAMVIG guidelines, we've decided to stick with the PS3, but we have actually increased the strengths. You can use PS3 up to very strong if you've got 2 orthogonal assays showing loss out of frame transcript and no evidence of leakiness. Next slide, please. And I'm now handing back to Trudi.
Thank you very much, Miranda. So continuing with the deep dive into the classification codes, we're moving on to PM2 and BS1 for population data. So PM2 rare in controls and BS1 common in controls. Next slide, please. For these, I'm going to look at 5 BRCA1/2 variants from previous runs. And in this table, you can see the NOMAD data, total NOMAD in the 3rd column, the 4th column, NOMAD data for, apologies, the 4th column for NOMAD data for in the non-cancer females cohort. And then in the final 2 columns, the number of applicants, number of participants who applied PM2, the total number, and in brackets you have the percentage, and the number of participants who applied BS1. So next slide, please. So what do the guidelines say about PM2? So the original ACMG guidelines say that PM2 can be used at a moderate level if the variant is absent from large general race match population. The ACGS 2020 guidelines say that it can be used at moderate if it's absent, but also if it's rare in NOMAD by using scientific judgment and the CardioDB tool to help with this. The general cancer guidelines allow the use of PM2 at moderate or supporting level and give extra clarity so that it can be used as moderate for no observations in a cancer-free control series of more than 50,000 individuals, or it can be used at supporting if it isn't absent, but it's at a frequency of less than 0.002% in a cancer-free control series of greater than 50,000 individuals. The gene-specific guidelines for BRCA1 and 2 give extra guidance on this. Again, you can use PM2 at moderate or supporting, but the NOMAD data should be from cancer-free female controls due to the low penetrance in males. For CHEK2 and ATM, PM2 is only allowed to be used at a supporting level because of the common phenotype that's associated with these genes. So next slide, please. So in the examples that we've selected here, if you look at the number of cases in the non-female NOMAD cohort, the variant is present on NOMAD. So to see if PM2 applies at a supporting level, you can use you need to see if the threshold of less than 0.02% in greater than 50,000 individuals is reached. So you can see in all these cases in yellow, we don't reach that threshold. So therefore, in these examples, PM2 cannot be applied. So next slide, please. Looking at BS1, which is common in controls, again, all the guidelines say that it can be used at a strong level. And the ACMG, it advises that if the allele frequency in a control population is greater than expected for a rare Mendelian disorder, it can be used at strong. The ACGS 2020 advises using CardioDB to calculate if the numbers that you're seeing in NOMAD exceeds the maximum credible allele frequency for the disorder. Again, that's for rare diseases. The general cancer guidelines say it can be used at strong using CardioDB, and for BRCA1 and 2 specific guidelines, you can actually apply this maximum tolerated allele frequency figure that's been calculated for you at 0.001% and put this into CardioDB to be able to assess whether the numbers you're seeing on NOMAD qualify the use of BS1. And I'll show that in a little bit more detail after this. For CHEK2 and BSEP ATM guidelines, it can be used at strong with the threshold being greater than 0.05%. So next slide, please. So an application of using the CardioDB tool can be seen here. So in yellow to the left, you can see the NOMAD non-cancer female data set selected. And here you can see that for this variant BRCA2, C1460C2A, which is our first variant that we're looking at, there are a total of 6 female cases in 52,887 individuals on the NOMAD data in the control database. So knowing that the maximum tolerated allele frequency for BRCA1, BRCA2 and PALB2 is 0.001, this can be directly put into the CardioDB tool under the tab Calculate AC, Calculate allele count. So you put that in the first box, and then in the second box you put in your data from NOMAD, and this will give you the maximum tolerated reference allele count of 16. So basically, if the numbers you're seeing on NOMAD are greater than 16 for this variant, then BS1 can apply, but if it's less, BS1 does not apply. So next slide, please. So looking at our examples in a bit more detail then, the third column from the right-hand side, the MTAC, calculating this using the CardioDB, you can see the maximum tolerated allele count for each of those examples. So if you directly compare that to the numbers that you're seeing on the non-cancer female NOMAD cohort, you can see that none of these reach the threshold to be able to apply BS1. So for these examples, neither PM2 nor BS1 apply. So next slide, please. So looking now at PS4, which is your case counting or case control. Next slide, please. What the guidelines say about PS4 case counting, the ACMG say that PS4 can be used at a moderate level if you are seeing the variant in multiple unrelated patients with the same phenotype and it is absent in controls. The ACGS 2020 say that it can be used at moderate and supporting depending on the number of cases that you're seeing. So if you're seeing 2 or more unrelated unaffected individuals, you can use it at a moderate level, or if you're seeing 1 unaffected, unrelated individual and it hasn't been reported on NOMAD, then you can use PS4 at a supporting level. The general cancer guidelines also follow the ACGS guidelines. And then there's a recent update an update to the BRCA1 and 2 guidelines follow the general guidelines that give extra guidance because of the common phenotype associated with these genes. So extra stringency is applied so that PS4 supporting can be applied if you're seeing the variant in 5 or more different HPOC families, and it can be used at moderate for more or equal to 10 different HPOC families, and also that PM2 has not been applied. For CHEK2 and ATM, these recent guidelines do not allow the use of PS4 for case counting because of the common phenotype issue. So next slide, please. So if you're case counting, you need to make sure the variant is absent from NOMAD, that the phenotype is consistent with HBOC, and sources of cases that you can use for case counting are peer-reviewed scientific literature, inter or intra-diagnostic laboratory databases, curated databases such as LOVD and HDMD, but not ClinVar unless you can actually obtain detailed information on the phenotype of the cases. So next slide, please. So where PS4 is really useful is in case control analysis. And here you can confidently use it at different levels because you are obtaining a statistic, the significant results when you compare cases and controls. So your cases and your controls must be ethnically matched. An example of where you can obtain cases information is in the CanVar UK database, which is accessible from the CanVig website. It's an information resource where all the data from genetically tested cancer genes from UK laboratories is deposited. So you've got cases together with all the numbers that have been screened. And then you can use this and compare with controls on NOMAD using the tool that's detailed there, the Stat Pages tool. So next slide, please. So as an example, looking at this BRCA2 variant, if you go onto the CanVig CanVar database, you can see here that there are 3 cases where this variant has been identified in the testing cohorts from the UK laboratories, in a total of 56,427 total probands tested. You can use your NFE equivalent from the white ethnicity cohort to give you 3 in 43769. And then you can compare that to the NOMAD figures if you look to the right using the NFE data where there's 1 case in 51,349 individuals sequenced. So you can put that into the table in the Stat Pages tool. On the left, where your condition is present, that's your cases. So that would be 3 in 43769 minus your 3. And then for your controls, you put it in the absence section. So it's 1 case in 51349 minus your 1 case, which is 51348. Then this will calculate your p-value. And in this case, it gives you 0.340. And you can see by looking at the table associated with the PS4 that in this case it doesn't reach the threshold for being able to use PS4 even at a supporting level. So in this case, PS4 cannot be applied. Next slide, please. So at the other end of the scale then, an example is this BRCA2 variant. Again, on the CanVar UK database, you can see there are 19 cases reported. And in NOMAD, there are 2 cases. So again, putting this data into your statistic tool, you obtain a p-value of 0. Now, the odds ratio is really important here because the cases on the UK database include a lot of historical cases that would have been ascertained when family history information was critical, and there was strict criteria for selection for testing. So therefore, we consider it enriched familial cases, and the OR is therefore set at stringent greater than 10 and lower 95% confidence intervals of greater than 1. So you can see that these have been reached for this variant, and this result allows you to use PS4 at a very strong level for this particular variant. So next slide, please. Case control information can also be available in the literature. So in run 9 variant, this CHEK2 variant was one of the cases involving a deletion of exon 9 to 10. And in this 2007 paper, it's presented to be present in 0.4% of the controls and in 1% of unselected breast cancer cases, giving you a significant p-value of 0.02 and allowing PS4 to be used at a strong level. So next slide, please. So now we're going to move on to the PP5, BP6 reputable source codes. So this is codes that can be applied to data that's reported on the ClinVar database. Next slide, please. The guidelines say initially ACMG rules say that these codes could be used at a supporting level. The ACGS 2020 guidelines removed the use of this code for rare diseases, but allowed an exception for common cancer susceptibility genes due to the large data sets that are available on the ClinVar website, data from Ambry, Invitae, GeneDx, for example. However, the updated cancer susceptibility genes guidelines that came out in December 2022 have removed the use of this code under the exception rule based on the Biesecker and Harrison SVI recommendations. The BRCA1 and 2 2022 guidelines will be following this recommendation very shortly, but at the moment in this guideline that's still available to refer to, the exception rule is still allowed, whereby these two codes can be used at a supporting level. And also in here, there's guidance on how to apply Enigma multifactorial data to be able to use PP5 and BP6 at either strong, moderate or supporting. And I'll go into that in a little bit more detail in the next few slides. The recent guidelines on ATM and CHEK2 have removed these codes. They are no longer allowed to be used for this. Next slide, please. So looking at these codes in a bit more detail, in run 6, we had this variant where the expected outcome was a class 2, but where most of the participants returned a class 1. Now this is likely because of the entry on ClinVar, the 3-star entry of benign based on the Enigma expert panel classification. And you can see many of the class 1 participants applied BP6 at very strong and strong, likely based on this Enigma results. So this would be an inappropriate use of BP6 according to all the guidelines. So using BP6 in this way, the maximum strength it could be used is BP6 supporting, giving you an overall classification of 2. Next slide, please. But since run 6, recommendations, the CanVig BRCA1 and 2 guidelines have provided guidance on how to extract information from the multifactorial data. And on the CanVar UK website, it's very helpful information on the various epidemiology studies and the likelihood ratios and the conversion to exponent points to help you apply the multifactorial data. So in this example here, you can see that in the Parsons paper, if you look at the combined likelihood ratios, you get a conversion figure of -32.4. So if you're using this data, the combined data, it allows you to use BP6 at a strong level. If you look at the table there, the -32 is much lower than the -4 at the strong level. So you can use BP6 at strong, or alternatively, you can use the individual components. So you can, for example, use the family history and pathology together to apply to BP5 at a strong level. And you can also use the co-occurrence to apply to BP2 at a supporting level. Again, reading the exponent figures off the table. So next slide, please. Similarly for PP5, this variant in run 8, for this variant, there was a 3-star ClinVar entry of pathogenic, again from the Enigma expert group. And again, PP5 was used at a variety of strengths, but using it on the based just on the ClinVar evidence, it should only be used at a supporting level. But again, extracting the multifactorial data, you can see here in the IRC Valley information, there is information on the segregation, a likelihood ratio of 229, converting to an exponent score of 7.42, which allows you to use PP5, which allows you to use multifactorial data under the PP1 score code of strong, which is the segregation code. It allows you to use it at strong. So next slide, please. Now moving on to the PP3, BP4 in silico evidence. Next slide, tools, please. So the guidance initially, all of them allow it to be used only at a supporting level as maximum strength. It's for missense variants, and the ACMG suggested commonly used in silico tools, polyphen, SIFT and mutation taster, and it provided in table 2 a list of common splice prediction tools that could be used. In the ACGS, they again used these same tools, but indicated that there would be meta-predictor tools coming shortly that probably would overtake these. And also for splicing, recommended the use of Maxent scan and SSFL at designated thresholds indicated there. The general cancer guidelines again use it supporting, but now 2 years after the ACGS 2020, REVEL is the recommended protein impact tool to use, and the use of multiple tools is no longer recommended. You can see the thresholds there for PP3 and for BP4, and if your variant doesn't reach these thresholds, then these codes cannot be used. For splicing impact, Splice AI or Maxent at those thresholds indicated there can be applied. And the BRCA1 and 2 specific guidelines follows the general cancer guidelines, and then for CHEK2 and ATM, the slightly different scores for the REVEL thresholds. Next slide, please. So now handing over back to Miranda to talk a little bit about when conservation needs to be considered.
Thank you so much, Trudi. So yeah, this is the last little bit on the codes. I know it's kind of, we've given you quite a lot of information. I hope it's been really useful. So some of the codes where you might need to consider the conservation and take that into account is PM4 and BP3 for in-frame deletions or insertions, or if it's a BP7 synonymous variant. Next slide, please. So PM4 and BP3, this is from the CANVIG guideline, and we're saying that it should be applied with caution in poorly conserved regions. At the moment, we're saying that they've given you some examples of in silico tools that can help support your decision to apply PM4. In the fullness of time, we're hoping that there's going to be, big studies on these in silico tools and really kind of work out what's their sensitivity, specificity, negative predictive value, etc., and be able to recommend which tools kind of are best to use. But at the moment, they're quite limited. And we would recommend that you use it at moderate if it's an insertion of deletion of 2 amino acids or more. But if it's only a single amino acid, you're better off using that at maximum strength of supporting. Next slide, please. So looking at this example, BRCA variant, this was a single amino acid deletion of asparagine 369del. We were expecting the classification to be class 1, but you can see that there are a significant number of participants that scored it as class 2 and class 3. And in fact, PM4 was applied by 36.5% of participants. Next slide, please. So if we have a look at this region, this is on Alamut Visual Plus. So on the kind of left top hand side, you can see that it's this variant is not in a functional domain. The variant is only found in mammals. It is conserved in mammals, but it doesn't kind of go any further down the kind of evolutionary tree. On the left-hand side bottom is a couple of examples of the output from the in silico tools, which are fairly borderline. I don't think kind of really support the use of PM4 or BP3. And then again, looking at the nucleotide conservation, this is a screenshot from Alamut Visual Plus again, and kind of the top track shows you your nucleotide conservation, and you can look at your PLASCON score or your PILO-P score, and they're the cutoffs that have been recommended by ClinGen to define that. So you can see that this region is pretty poorly conserved also at the nucleotide level, therefore PM4 would not be appropriate. Next slide, please. So moving on to BP7, which is for synonymous variants. And so the key thing is here, if you have a synonymous variant, you have to check whether that and make sure there is no impact on splicing and the nucleotide is not highly conserved. In addition, BP7 can be used for two other examples. So it can now, and this is kind of standard across the ClinGen VSEPs, it can be used for intronic variants at or beyond +7 or -21. So don't use it for intronic variants around those kind of critical splice regions. And you can also use it for non-coding variants in the UTRs. I've got two examples here. So on the left is a BRCA1 synonymous change, 4185G to A. But you can see that this variant affects the last G of the exon. You can see that the nucleotide conservation on the track above is very highly conserved, and if you look at splicing, you can say that this variant is predicted to affect splicing. Therefore, BP7 is not applicable for this variant. However, on the right hand side, the BRCA1 C.38378 to C, you can see it's synonymous change and that the PLAS cons and the PILO P scores are very low. You can see from the graphic it's not conserved, and therefore BP7 would be applicable in this case. Next slide, please. So that's us done for looking at the individual codes, but we know one of the other really difficult areas is how you combine the codes. And most importantly, it's taking care not to use the same evidence twice in multiple codes. Next slide, please. So one of the most common ways that you can kind of overcount the evidence is some of these prediction codes. So PP3, PM1, PM5, particularly PM1 and PM5. So PM1 means your variant is located in a mutational hotspot or a critical, well-established functional domain. PM5 means you've seen a missense change, but it's the same residue where a different missense change has been determined to be pathogenic before. As you can see, these criteria are not independent and there is a significant overlap. So a mutational hotspot kind of by definition means there's highly likely to have missense changes at that same position. In addition, the CANVIG guideline sets out that you cannot use PM5 where you have functional data towards benignity. This is because your PM1 and PM5 codes and even your PP3 codes are all kind of prediction, computational, indirect evidence, whereas if you have functional data on that variant, that supersedes it because that is direct evidence of pathogenicity. Next slide, please. So looking at the example from run 6, this is a BRCA1 missense variant, P.glycine 1788 asparagine. We were expecting a classification of class 3, which the majority of participants did, but you can see that we've still got, nearly 120 participants scoring it as class 4 or class 5 with those significant clinical implications. Next slide, please. And actually, in the assessment of this case, 30% of participants used both PM1 moderate and PM5 moderate together. So that's the highest strength of both of those codes, and all of them classified it as class 4 or 5. If we look at this snapshot, this is the latest classification on Canva UK, that you can see kind of where it says data included in classification, there is a reasonable amount of evidence towards pathogenicity or the P codes, but then there is the Parsons et al. Enigma multifactorial data that actually has a strong likelihood ratio towards benignity, which would be sufficient to apply BP6 strong. And also, really importantly, where it says data not included in classification, you can see all those functional studies that were available at the time, obviously, of this GTAC run that show that it actually has fairly standard HR activity, and that most of these functional assays had classified it as functional or a VUS. And so again, this is much more important data than that kind of predictional data, and you would not want to be applying PM1 or PM5 for a variant that has got this much data from that direct functional evidence. Next slide, please. Another code where it can be tricky is for splicing, where you shouldn't use the BS3, BP4 together and PS3 and PP3 together. This can be a little confusing because for a missense change, we do allow these codes together because they are different pieces of evidence. So for a missense change, the in silico tools are looking at evolutionary conservation, and then your functional assay is looking at function. Whereas for splicing, the in silico tools are just predicting whether there is going to be an effect on splicing, which is then generally confirmed by the RNA studies. So again, we can use the splicing in silico tools to support the application of kind of BS3 or PS3, but not apply them together. Next slide, please. So example here is run 6, C.301 plus 7, G to A. We were expecting classification of class 2 and majority scored it as class 1. Thankfully, this one, there's no clinical impact. Next slide, please. And then looking at a different variant, we've actually looked at this variant before, this BRCA2 variant at -5. Again, there was a bit of a spread. We're expecting class 4. We saw class 3 and class 5. Next slide, please. And then having a look at both of these variants together. So you can see for the BRCA1 301 plus 7, the vast majority of participants that scored it as class 1 used both BS3 and BP4 together. And for BRCA2 variant, the pathogenic one, again, when they scored it as class 5, so a bit overweight in the evidence, again, the vast majority of participants have used both PS3 and PP3 together. Next slide, please. So more good news is that a lot of this, I know this can be very difficult, but this is laid out really nicely in the CAMVIG guidelines. So if you go to the CAMVIG guidelines and go all the way to the end of the document, there is this fantastic grid that shows you all the different kind of code combinations. So where you've got the pale green, that means it's fairly straightforward. You are allowed to use those codes together. Where it's a darker green, then it can be permitted, but there might be some caveats. And so there's a little number and you refer to the table on the right hand side to make sure that you are applying it appropriately. Red means is not permitted to use these codes together. And again, some of them have the code. And so again, on the right hand side, I've just highlighted the PM1 and the PM5, showing that is not permitted. Next slide, please. And again, we've got exactly the same for combining all the benign criteria. And again, I have highlighted the BS3 and BP4 for splicing. Next slide, please. And then I'm now finally going to hand back to Trudi, who's going to talk about collation of codes.
Thanks, Miranda. So we've come to the final part of the webinar. And at this point, you've collected all your evidence, you've assigned all your codes, and you're at the point now where you're going to just add them all up, see what you have to obtain a final classification. And we have found, when we did our look back, that there are a significant number of cases where the final classification, even though the evidence criteria was correct, the final classification ended up being wrong due to errors in the summation. So next slide, please. So the initial guidance on collating your evidence is as given here on the original ACMG guidelines, where you can see they provide rules on combining the classification for pathogenic, likely pathogenic, benign, likely benign, and uncertain significance, and the various categories here. So next slide, please. So following on from that, the Bayesian classification framework was developed to refine this more, and as a result of that, the ACGS further refined the original rules to adjusting to the Bayesian framework to provide the table that's available for you to refer to in the 2020 guidelines. There's a small error here in that the cut and paste is just missed off the top section, but if you look in this paper, the top section should be pathogenic A, and the first line will be one very strong, and following on then a strong, moderate, and supporting. The CANVIG guidelines for the general cancer susceptibility genes also provide a very helpful numerical way of collating your evidence. So you can assign exponent points for evidence towards pathogenicity. So if you have a very strong piece of evidence, you can assign an 8. If you have a strong 4, moderate is 2, and supporting is 1. Or for benignity, if you have a strong, it's -4, moderate is -2, and supporting is -1. And the thresholds are if you have 10 or more, then you reach pathogenic. If it's between 6 and 9, it's likely pathogenic. Between -1 and -5, it's likely benign, and less than -6 is benign. And if you have a value that's 0 to 5, then that would be in the class 3 VUS range. So next slide, please. So in summary, what we've covered in this webinar, we've carried out just a review of what guidelines are available for participants to refer to as they go into Run 10. We've explained how they evolve over time and provided guidance on appropriate ones to refer to. We've covered an overview of previous runs, looking at tricky variants and codes that were observed, and in particular those where different outcomes could have had a clinical impact. And we've carried out a review of code strengths and combinations that we observed to be applied insignificantly by significant numbers. And we hope that we've provided some clarification to help with this for future runs. So thank you. Handing back to Simon and Sandi.
Thank you, Trudi. So we, I think, have been overwhelmed by the amount of educational content there. Fantastic. And I think you definitely have brought some clarity. I'm sure there'll be quite a few people who want to review it once and watch it again once it's available on the YouTube channel. So thank you very much indeed, both of you, because I know a huge amount of work has gone into that presentation. What we would like to do now is just advertise the fact that Run 10 is open from today onwards, and that will be open for your own convenience to log in and enrol and participate in the new run. The run will close on the 14th of April, and I believe we have about 450 individuals currently enrolled. So I think it's definitely a great opportunity to use the newly learned knowledge that we've just shared on this webinar to take that forward. So just please do get in touch with either EMQN's office or GenQA's office by email or on our website to find out further details of how you can enrol and for participant instructions, and we hope that you find this a useful exercise. We do plan to hold a second webinar following the close and the analysis of the data of Run 10, and I hope you can join us on that one. But now we have some quite few interesting deep questions submitted on the Q&A for us to now review. So Simon, will we kick off the Q&A now?
Yeah, we will. Thank you, Sandi. And obviously my thanks also go to Miranda and Trudi for a fantastic presentation. I think, you know, in the last sort of hour and 10 minutes, we've covered a huge amount of knowledge associated with it, and hopefully it will help you guys to move forward your variant classification. So we've got some questions. I'm going to start off actually just with a sort of general one really before we get into some more of the deeper stuff. But I mean, the ACMG guidelines were now 8 years old, and they were obviously one of the first set of guidelines to help laboratories classify these types of variants, particularly in the germline setting. And I presume that the guidelines that are due out this year will be more of an evolution rather than a revolution to those 2015 guidelines. Do either of you know in advance what the main changes will be or themes that will be coming from those new guidelines at all?
I've got a reasonable idea, and I'm afraid it's going to be a revolution. The codes are completely changing. Right. It will be all evidence points based. It will be in a lot of ways very, easier. So we won't have the kind of PP3, PS4, etc. It'll say in silico tools, or it'll kind of be a bit more obvious. You won't need to, the name kind of the evidence will be in the title. But yeah, there's still a lot of work to do, and they're not really expecting the guidelines to be out towards the end of the year, but there will be significant changes. So and a lot less codes, but yeah, they won't quite be the same as code. So it is going to be a big change for all involved.
Okay, so sorry,
I'm just going to follow up and say, so will there be more aligned to the copy number variant classification system that is based on figures and more evidence-based?
Yeah, I mean, it's kind of the ACMG already aligns to the Bayesian point system with the Taptigian paper, and so the plan is they're not including the CMV guidelines, and the CMV guidelines use slightly different weighting, yeah, between kind of lightly pathogenic and pathogenic, then it gets a bit different. But the plan is to publish this SMV guidelines and then align with the CMV guidelines, as far as I'm aware.
Thank you.
Fabulous. So I mean, I think what that's highlighting obviously is that, I mean, by the sounds of it, the publication is going to be the back end of the year, so it's probably going to be not effective for us to include them in this year's runs. Probably run 11 will still, although that's towards the back end of the year, may miss that publication date. Obviously we don't know when that will be, but clearly there's going to be a lot more learning required for every laboratory, which we'll try to pick up as obviously in next year's runs, and if that publication's out so late in the year. But clearly there's definitely a bit more work in terms of education and experience required for the laboratory to pick it up. Yeah, so just to feedback on, because of that, then we've had some discussions kind of with the ACGS, kind of covering all of rare disease, and the CAMVIG group as well, and we've agreed, because we know that this big change is coming, we're not going to make big changes to those guidelines for SMVs. For example, we're still using PM2 at moderate. There's the new PP3 paper, but we're not going to make those big kind of changes, because we know that we're going to have to then change everything towards the end of the year. So it felt like we'd be better off waiting, having everything together, and then making all those changes. Fabulous. So I think we've emphasised that also in the flyer this year, that we've made strong recommendations to you as the users of this service that we're providing, is that we're still following those original ACMG papers, plus the guidance as we've detailed earlier on in the webinar. So we won't be pushing the new ones forward until obviously next year, by the sounds of it. So I guess to the first of our sort of user questions, to go into a bit more detail, Miranda, there was a question there relating to strengths of PS3 and BS3, and also whether the new guidelines will include BP5 and PP5. Were you okay to take that as a question? Yeah, sure. So as I was saying, the BP5, PP5 will go. It has gone from those original ones, so I don't expect we'll see anything back, but it's all going to be so different, so we'll have to wait and see for that. In terms of PS3 and BS3, for the Finlay assay is classified as strong, and then we're still using the original kind of assumption in the Richards et al. Paper that we always have to have two pieces of evidence towards pathogenicity, so you can't use that as a standalone. So even PVS1 at a very strong is not used at a standalone, and the only exception is the BA1 category that's used as standalone. But yes, by all means, we have been through the Finlay paper according to Brunich, and happy for it to be used as strength strong. Fabulous. Sandi, do you want to take the next question? Can I just say, sorry, I just realised, going back to it, we're talking about the ACMG updates, but what we do know is coming this year is the Enigma VSEP guidance. So that has gone through, and that is a lot of, some significant changes. So we will have to think about, depending when the publication of that is, how we then incorporate that into GTAC runs, because for assessors, we're trying to be very flexible and open to whatever classification system you're using, but it can be quite hard work for us to classify variants to multiple different guidelines. But yeah, that one is likely to be out much sooner than the ACMG.
Thanks, Miranda. That's good to know. We'll keep everyone informed on that. Yes, Simon, there's a couple of themes coming up here. I'm going to just pick out software and pipelines and automation of variant classification. I think that's something that we're all struggling with, just the amount of workload that actually involves with cases in the lab. So somebody's asking, is there any validated software or pipelines that can be used for automatic variant classification? And following on, there's various commercial databases out there that can provide the codes and give you your final classification. Can we still use those commercial services? How do we know that their database is up to date and they're applying them correctly? What's your views on that?
Miranda, you might be the best one for this if you have some exposure to it.
I wish there was. It's my very short answer. I've tried a few of them, and there's still a lot of work to be done. I think from my experience, lots of them overclassify variants. They particularly overweight PM1 and kind of other of the prediction codes. And yeah, I don't know any that are suitable for use as they are. There are kind of commercial companies and things that are focusing on variant classification, and when you've got a kind of human looking kind of partly at AI and managing that, then they may be better. But yeah, for now, I think that there's just, and then as we've seen, there's just a lot of complexity involved in variant classification, different strengths, and then how you combine the evidence and how you not making sure you're not double counting everything together as well. So I'm afraid it is still quite a manual process.
So plenty tools out there to help us.
Yeah.
But I think we're never going to get away from that individual review and judgment. And
things like even like a ClinVar 3-star rating. We did, Claire Turnbull raised the question at a conference to Stephen Harrison whether we could just take that says that if there's a 3-star rating as pathogenic, and actually he said no, really, that kind of that body that's got that 3-star rating should provide the evidence, and then you should combine that with what evidence you've got to make sure you agree, because you might have some evidence suddenly that contradicts it, or you might have some additional evidence that increases that strength. And so really we should all be looking independently for kind of governance and kind of taking that evidence but also using our own data.
It evolves. We're all learning, aren't we? Simon, is there anything you want to pick up?
Yeah, there's a couple more questions, Sandy, I'm going to pick up on. So I mean, one of them is actually more of a comment than anything else. So one attendee has asked whether they can receive slides from the webinar prior to the recording. I presume that actually means post-recording. We haven't traditionally provided the slides. Obviously, we provide the full video, the full webinar as a video for users to watch back at their leisure. We'll look at that whether that's an option for us. Obviously, there's some reasons why that may or may not be so easy to do, but we'll certainly look at that. So that's possibly something we may be able to facilitate in future, but certainly the webinar will be available to all users via the GenQA and EMQN websites post that. There's more of one more specific question that's come through is relating to the ClinGen group. So does the ClinGen group not recommend, so doesn't recommend the use of PS3 and BS3 for the PALB2 gene? Is anybody able to pick that up at all?
Miranda, are you okay with that?
Yeah, those guidelines aren't available, but so they are still in draft. But I know that we had this discussion in CanVig and said, can we use these because there aren't any pathogenic missense variants and kind of included in that Boonan study, and then the VSEP kind of was contacted and they said they're currently not using PS3 and BS3. As I said, that might change before publication. So we, as assessors for that variant, we agreed that it probably would be safer not to apply PS3, BS3, but I can't, I'm not in that VSEP, so I can't say what's going to be in the final published version.
Fabulous. Okay, Sandy, over to you. Do you think there's any more we can go through now, or we good to handle those through?
Yeah, I think we're getting lots coming in. So Simon said we will definitely respond in the EQA summary report after run 10. But there's one here just for clarification. So if you have a synonymous variant that's located at the last nucleotide of an exon, and it's predicted to result in exon skipping leading to a frame shift, can you use PVS1?
And they actually provide a case as well for that one, I think, as well. The BRCA2 variant,
So yeah, at the moment, the last G of the exon and the +5 GTA, which we know from kind of publication again, is very highly likely to affect splicing and not included within PVS1. I hoped that the splicing SVI paper that is actually out as a preprint now would include those, but because they haven't done what they said was that because they haven't looked at the other variants around it to know whether it was kind of in frame or out of frame, and you still got to do that assessment. They haven't included those. But for example, the APC VSEP are using the last G of the exon and are applying PVS1 from supporting, I think, from not applicable up to strong for some of those. They've assessed each of those last exon variants. They're variable, but currently that, as far as I know, is the only VSIP that's that I'm involved in that is using PVS1 for those last exons. So currently no, all you can apply is a PP3 supporting, whereas sometimes it's a bit frustrating because all the in silico tools are showing a complete loss, and you want to apply that at higher strength, but no, not currently.
Thanks, Miranda. That was very, very pertinent question to ask. Thank you.
Got one more, Sandi, relating to PS3. So to the panelists, what's your point of view concerning the variants classified PS3 by Lyra et al. for BRCA? I think that must be BRCA1 and 2. Do you set these as PS3V strong? Too specific, i.e. the paper itself?
I haven't assessed that, but I remember when we were doing the Finlay as well, we combined the Finlay and the Bowman to see if both together you could reach, and you couldn't get to a very strong. So you really do need, I can't remember there's over 120 variants in Finlay alone, and plus another, so that's kind of 200 plus variants. It's all to do that odds of pathogenicity. You just need to go and calculate it, but you really do need a lot of known pathogenic and known benign variants. Not, this is really important, is not kind of using those the VUSs in there to calculate that score.
Fabulous. I think that so far that's all the questions we've got, Sandi. I think we've covered all of those.
And great questions. Thank you.
Really good questions.
Indeed, and for Miranda and Trudi dealing with them live with no preempting. Very challenging sometimes.
Absolutely, completely agree. We've got we're sort of 5 minutes before we're due to call it a day, but in the absence, I think, of any more questions, what we'll do is we will wrap it up now. As we've talked about before, if there are any more questions that come through, I mean, please use our feedback survey that we talked about, and that you will get that link later on, just after this webinar. If there is anything else, we will of course cover it in the report for that run. So please do try to join us for run 10, and obviously later on in the year for run 11. Each of those webinars will be we'll receive a webinar afterwards summarizing what went on. Can't get my words out today. So I think what we'll do is we'll call it to the end of day. I'd just like to thank the speakers, please, to Trudi, obviously, and Miranda for a fantastic presentation. I think you'll all agree it's really, really educational. I'd like to thank my co-host, Sandi, and most important, I'd obviously like to thank you guys, the participants who've dialed in today, over 230, I think, and 300 plus registered as well. So fantastic participation. We look forward to you joining us in run 10. And as we said before, we'll put this webinar, we'll make it available from the GenQA and EMQN YouTube channels sometime in the next couple of weeks. We'll let you know when that's available. So thanks once again, and have a good rest of the day.
Thanks, everyone. Bye.
Thank you.
Thank you.